uh, tutorial for the next minutes. I have a few questions, but first I would like to know whether you have any comments or questions to the lectures I have given the last few days. Oops. All right. Okay. Now Bruce is here. He will probably force you to have some questions. Any comments, questions, or stupid questions are most welcome. It's usual. If not, then maybe I should start. So there was a question yesterday about do we need these population cycles for balancing selection? It was a question from up here somewhere. You, yeah. Well, so maybe let's look a little bit what happens under which circumstances you get these population cycles. So sometimes you get just a stable equilibrium. Let's suppose this is inside such a phase plane, and where one axis is the frequency of the virulence allele in the parasite, and the other one is the frequency of the resistance allele in the host. And here the resistance allele. And if you have, in the deterministic model, so no drift or so is involved, a stable equilibrium, and if this is in the interior, not at the edge also, yeah, then you have an example of a balancing selection. Okay, you don't need anything else. However, it can, if it is possible that this equilibrium point is unstable, yeah, then you end up with cycles that are, or with cycles that are going around here. Yeah, they can be limit cycles, yeah, that are stable, or they can also be cycles such that if you plot it on with respect to time, that they show a damped oscillation and they end up in this equilibrium point. Okay, something like that. So in other words, you don't need cycles to have balancing selection, but you need negative frequency dependent. The mechanism needs to be negative frequency dependent, include negative frequency dependency, otherwise you wouldn't get that. If you have, however, a stochastic description of this problem, then you don't have this stable equilibria, because the drift always notches the trajectory away from the equilibrium point. Yeah? And so that means you have then this fluctuations around this equilibrium point. Yeah? This is due to drift. In a deterministic system, you would end up in that equilibrium point, but not if you have random genetic drift. And now this word trench warfare usually means something like that you have small advances and then again a return also. This is not these big cycles, at least where the word is coming from the military, you know. So what happened in the First World War, this was what the Germans and the French and so what they did, for example, in that region, in this border region between France and Germany, that is called trench warfare. Yeah, so little advances and recess, little advances and recess. Okay, that is trench warfare. It's not this big boom and bust cycles. So these are called boom and bust cycles if they almost go over the whole phase plane. Does this answer your question better than yesterday? But at least that is the dynamics, you know. If you look at the dynamics, the dynamical system, and you study this behavior in a qualitative way, that is what you get out. All right. Any other problem we should discuss again? If not, then I have two problems for you to work on. One is this one. So this is a result of a genome scan where we scanned all chromosome arms of Drosophila, so the X chromosome of Drosophila melanogaster, the X chromosome, the second chromosome, the left arm and the right arm, and also the third chromosome, the left and the right arm. Yeah. And the scan, as I mentioned yesterday briefly, was done such, if this is a chromosome and here is the centromere, it was done such that you move windows of a certain size across the chromosome with no overlap. So the first window size was 200 and 500, 1,000, 2,000 and 5,000. Okay. And doing this, we covered the whole region of the chromosome, except those regions which are in very low recombination rate near the centromere. Yeah? Because these regions are not very well sequenced. This is repetitive DNA, which is hard to sequence. Okay? So you can say that the DNA, what we looked at, is the so-called euchromatic region, where the genes are. Yeah? In the heterochromatic regions, there are almost no genes. Okay, so now this is the result of this scan. You see that this chromosome here, which is 3R, shows the steepest decay going from windows of 200 to 5,000. Okay, so It goes up here, they're all nearly identical, and this goes down very steeply from 200 to 5,000 base pairs. Oh my God, this is strange. Pair bars or something like that. Oh well, what can I say? Um, then the next chromosome arm, which is marked by these triangles, this is the left side or the left arm of chromosome 3 is also going down. Yeah, so up to here. Then the next one is 2L goes down. Okay. However, what's interesting is that the X chromosome and also 2R, they are almost constant. Well, what we got out of what we looked in this scan, we looked at the top, whatever, 5% of the windows that have the largest, the top 5% of the largest theta values and the largest Tajima D values. Yeah. So both of the proportion here, which is shown here, is intersection of the two data sets. Yeah, so the data sets have to fulfill both requirements. Okay. This is exactly my question for you. Yeah. How big? Ooh, so in total, I think it's 120 megabases divided by 5. You have 5 arms, they are almost all the same size. So 25 megabases each. You see some, in this. Yeah, this is interesting, yeah. But why? I mean, I was very happy when I saw this. There must be a reason. What did I talk about? I think since Wednesday I talked nothing else but hitchhiking. Why is it hitchhiking? It has something to do with hitchhiking. Now you have to combine what I said about what determines the hitchhiking effect. Sorry? Wonderful. Yeah. So the hitchhiking effect is determined by, essentially by this parameter. recombination rate divided by S. S is the selection coefficient of this beneficial mutation, and R is the parameter between the selected site. Oh my. The parameter, the recombination rate between the selected site and the neutral site. Yeah. So if you have a smaller fragment, then of course the probability that there is a selected site in that is pretty low compared to a size of 5,000. Okay, so this is one thing. Now, why are then, this explains something, okay, but it doesn't explain everything. Why are these X chromosome and 2LY not, and 2RY, they are less steeply declining? Sorry? So apparently there is more recombination. And this is a well-known fact that the X chromosome shows a so-called X effect. Yeah. There is more recombination on the X. The X chromosome has more recombination. That's the reason why you see large X effect. The strongest adaptation in Drosophila happens on the X. Okay, so this is a well-known fact. Sorry. No, it doesn't have to do with the. This is sort of factored in. Yeah. Okay, so this is a well-known fact. I was surprised that also 2R shows this effect. I didn't know that, but then I looked up the recombination profiles. Yeah, and for example, Josep Maria Comaron, he calculated, or he has very nice recombination profiles for all chromosome arms, and the profiles are not identical from chromosome arm to chromosome arm. Yeah. So let me draw a picture. Yeah. So if this is one chromosome arm, then it goes up, goes down again. Okay. Let's say this is one. This is the centromere, let's say, in the X, and this is the telomere. However, it can go up to different levels. This is one thing, so it can look like this. Okay, and also the width of this valley of reduced variation, of reduced recombination, is not identical in all chromosome arms. Yeah, so if you calculate then the average average recombination rate, yeah, so you average over all recombination rates with your window. So you look up for each window the recombination rate. You can do that in Drosophila, and then you average over the whole chromosome arm. Clearly, those upper two, have higher average recombination rate. Yeah, so there's a clear, I was surprised, it comes really out, okay? That these last three have a lower average recombination rate than the other ones. I have no biological reason why that is, Yeah, but it clearly, There's a clear correlation with average recombination rate. No. So that was published before that, you know. The recombination rate has been measured in Drosophila quite well. You know, there are lots and lots of crossing experiments. And now we have also these SNP data, and with SNP data you can then measure recombination rates by measuring linkage disequilibrium. So that these data are very solid. Okay, so the last aspect of this, yeah, there is hitchhiking going on quite clearly, but is it hitchhiking with positive or with purifying selection, with background selection? This is a hard question. Yeah. Sorry. Uh, Purifying selection, if purifying selection is not so strong as Brian Charlesworth claimed for background selection, then you see also negative Tachima D. So it has the same properties as positive directional selection. Yeah? But if you go into the Charlesworth parameter range, then it's a different story. Yeah? But here it could be both. I'm not claiming this is only positive directional selection, which makes these slopes here. Okay, I guess this slide sort of is a summary of what I talked about in the last 3 lectures or so. Yeah. Any more comments, questions? Speed of adaptation in asexuals. In asexuals. In sexuals. Yeah, that is it's the more recombination you have, this is the large X effect. Sorry? Yeah. So I would refer you to the papers by Nick Barton and several of his postdocs. There was one guy I remember he had some papers about that. I met in Berkeley, and he's now in Berkeley as a postdoc. Sorry? Weismann, yeah. Weismann, Daniel Weismann. Yeah, Mr. Chima D, no. We have a long time tried to distinguish um, background selection from positive direction selection using the Chima's D. It was not possible. Yeah. No, that was a failure for more than 5 years in my life. Yep. This is um, the recombination rate in Drosophila is only measured for females. Yeah. But if you are interested in that problem, there are species uh, of Drosophila that show male recombination, Drosophila ananaceae. Yeah. And why that is, and there's the Japanese groups working on that, and I think there are specific genes that determine that, but I'm not up to date on that. But it is possible. No? Yeah. Any other question? Okay, so then I have one last question. Um, this is again about balancing selection. So, and I mentioned that ADH. So ADH looked like this. If you plot here pi between, and here's the fast, slow polymorphism. Can you explain this? Yeah, the first one, as we already mentioned, um, is a derived allele. Yeah, so it occurred uh, after the slow one. The slow one was there. It's an ancestral allele. The first one is derived. So why do we see this hump? And it is really a significant effect. And it was then modeled later by, so first it was shown in that HKA test that I showed you. And then it was modeled later by Hudson and Kaplan. So they inferred the parameters of that model. Just an intuitive explanation. Sorry? The x-axis is a gene from the 5' end to the 3' end. So this is ADH. Okay, so here you have the fourth exon, and then you have a small intron, and here, something like that. Four exons. Sorry? The fast, slow polymorphism. So again, it is a hitchhiking effect. Does it help? So some think about how you would get um, a highly diverged region just around this selected site between the fast and the slow alleles. And I'm saying it is due to hitchhiking. And Away from that, it is normal. So in other words, why do the fast and the slow alleles diverge? They can only diverge by accumulation of mutations, right? So you have the slow alleles in the population, and then the mutation from slow to fast occurs. Okay, so at that particular point, the fast and the slow alleles are different. Yeah? If you would measure FSD, there would be FSD would be one, right? Completely diverged or differentiated. Okay, so let's say this is a fast allele where it occurs. Then you see here, I think here is oh, I forgot the A and the doesn't matter. Yeah, there's one nucleotide difference. At this exact position, they are different. So if you would measure pi between, that means you take one sequence from the fast subpopulation and the other one from the slow subpopulation, you see divergence. Yeah? The only question is, why does it extend and why does it not further extend? Okay, and this is because mutations accumulate after the fast allele occurred. They accumulate and they cannot go from one of these subpopulations to the other one. Yeah, they can only go by recombination. By recombination, close to that fast-slow polymorphism is not possible because it is too small, this region here. No recombination events or not enough happen to scramble this whole population. Okay, so therefore, just around this fast-slow polymorphism, you see a divergence of these two classes. Yeah, and as I told you the other day, this region here is only about 200 base pairs. Yeah? Everything else is chopped up. So you see that balancing selection can also lead to a hitchhiking effect, which you can observe. We did not see it in our genome scan, of course, because it is too small, this window. And we did not really look. If we would have looked between, oh, sorry. If we would have constructed the genome scan such that we have one subsample of fast alleles and one subsample of slow alleles, we should have seen it. But of course, that was not our goal. We took only a random sample from a local population. Yeah, that if we would have known this, okay, but we were not interested specifically to find this, although we should have probably tried it. But you cannot divide the whole. You have to do it for each gene, or fast and slow. Okay, then you have for each gene you have maybe a different division. Then. Yeah, but then you have made a genome scan for this one gene, which is not what we want. So there's no other way than to do the genome scan than what we did. Just take a random sample from a local population. Okay. So that brings me to an end here.